Right now, inside your body, there is this microscopic molecular machine that is unzipping your DNA at a speed of about 50 letters per second.
Which is just impossibly fast if you think about it.
Right. And it's making a perfect copy of the chemical recipe that keeps your cells functioning, your heart beating. I mean, it keeps you alive.
And we just completely take it for granted.
Totally. We rarely stop to think about that invisible machinery. But today we're looking at the ultimate rule book that teaches teenagers exactly how all of that works.
Yes.
Welcome to our deep dive. We aren't just looking at random, disconnected science facts today. We are exploring the exact blueprint of how a whole nation teaches the fundamental laws of life to the next generation.
It really is a remarkable document to explore. I mean, we are looking at the official Life Sciences Grade 12 Examination Guidelines 2021. This is from the Department of Basic Education in the Republic of South Africa.
Our mission today is to explore not just what information is deemed critical enough to test on a national scale, but, how this educational system expects young minds to process the very building blocks of existence.
Exactly, from the molecular level all the way up.
Right, from the microscopic DNA coiled in our cells all the way to the complex macro-level interconnectedness of human reproduction. Okay, let's unpack this. I am so incredibly excited to reverse engineer a national curriculum to find the most fascinating nuggets of biology.
It is a great exercise.
It's like we get to see the matrix of how scientific literacy is actually built,
Well, if we connect this to the bigger picture, an exam guideline isn't just a list of chores for students to complete. It acts as a mirror.
Oh, interesting. How so?
It's a direct reflection of what a society values in scientific literacy. There's this calm, structural beauty to these guidelines. They outline precisely how to build a scientifically literate citizen step by step, concept by concept.
Ensuring they actually understand it, not just parrot it back.
Precisely. Ensuring that students aren't just memorizing.
So, before we jump into the biology itself, the actual cells and organisms, we need to understand the mechanics of the exam.
The rules of the game.
Yes, because knowing the rules of the game tells us a massive amount about how these students are expected to think.
And it is quite a rigorous game.
It really is. The document lays out that the students face two massive papers, 2.5 hours each, 150 marks each.
And they are structured very deliberately.
Right. Section A is your rapid-fire round, short answers, multiple choice, terminology matching, and then Section B hits them with these comprehensive, multi-part questions.
It's an endurance test, undeniably. But fundamentally, it is a test of varied cognitive skills. The guidelines explicitly lay out 3 specific aims.
Right. Aim 1, Aim 2, and Aim 3.
Exactly. Specific Aim 1 is knowing the subject content. That's your standard foundational knowledge.
Facts.
Yep. Then specific Aim 2 is doing science. So practical work and investigations.
But the cognitive weightings, that's what really caught me off guard.
Oh, they are fascinating.
Only 40% of the exam is based on strict knowledge. The remaining 60% is completely divided up.
Right, into higher order skills.
Yeah, it's like 25% comprehension, 20% application, and 15% analysis, synthesis, and evaluation. Wait, so if I just memorize the textbook front to back every single Latin vocabulary word, I still might fail.
You very well might. You probably would, honestly.
That is wild to me.
And that brings us to specific Aim 3, which is perhaps the most fascinating part of the entire framework.
Okay, tell me about Aim 3.
So specific Aim 3 relates to understanding the applications of life sciences in everyday life, as well as understanding the history of scientific discoveries.
Which is huge.
It is. And crucially, the relationship between indigenous knowledge and science.
I mean, the stereotype of high school biology is just rote memorization, endless flashcards with unpronounceable names.
Sure, that's the old paradigm. What this curriculum is doing is demanding critical thinking.
By giving so much weight to the higher order stuff.
Right. By dedicating the majority of the cognitive weight to higher order thinking, and by explicitly integrating history and indigenous knowledge, the exam forces students to see science as a living, breathing cultural practice.
Can you give an example of the indigenous knowledge part?
Take traditional healing, for instance. A student might be asked to analyze how traditional healing practices utilizing specific local plants led modern pharmacologists to discover active compounds for new medicines.
Oh, wow. So bridging the gap between old wisdom and new science.
Exactly. It frames science not as magic handed down from a textbook, but as a process of observation and discovery that has been happening within human cultures for millennia.
And there's an incredibly specific difficulty filter built into this exam, too.
The level 7 questions.
Yeah. The guidelines state that 5% of the exam is reserved for very difficult questions. And the text actually says these are specifically designed to distinguish level 7 learners.
The extremely high-achieving learners.
Right, to separate them from the rest of the highly proficient students. They intentionally build a 5% gatekeeper right into the test.
It's a mechanism to identify true synthesis. Those level 7 questions aren't just obscure trivia facts.
Like, they aren't just asking for a harder vocabulary word.
No, not at all. They are complex, unfamiliar scenarios where a student must apply their biological knowledge in completely novel ways to solve a problem. They are hunting for the future innovators.
Okay, so we have the blueprint for how the students' minds are tested. Let's look at the ultimate blueprint for the students' bodies.
Moving into paper 2.
Exactly. The foundational topic that anchors this biology curriculum is DNA, the code of life.
A very hefty chunk of marks there, too.
27 marks. And I love that the curriculum hits specific aim 3 right out of the gate. It doesn't just start with the chemical structure. It starts with the history of the discovery.
Right. They need to know Watson, Crick, Franklin, and Wilkins.
Yes. They are learning that this invisible code had to be painfully cracked by flawed, brilliant human beings.
And once they grasp that human element, they dive into the molecular elegance of the structure itself, the double helix.
It's just so beautiful.
It is. The fact that the incredible, staggering complexity of all living things is built on just 3 basic components, really. A sugar portion, a phosphate portion, and nitrogenous bases.
Those make up the nucleotides, right?
Correct. And those bases are linked down the middle by weak hydrogen bonds.
Oh, wait, why weak? Doesn't DNA need to be super stable?
The weakness of those bonds is actually a crucial design feature. They need to be weak so the DNA strand can easily unzip when it's time to make a copy.
Oh, like a zipper on a jacket.
Exactly like a zipper. If the bonds were too strong, the cell could never replicate its DNA.
That makes so much sense. And then you have the famous letters, adenine, thymine, cytosine, and guanine.
Base pairings.
Right. A always pairs with TG, always pairs with C. It is the ultimate binary code, but with 4 variables instead of 2.
And the curriculum requires them to not just know DNA, but to fully understand its hardworking counterpart, RNA.
Which is like the slightly different sibling.
Right. Very much so.
Right.
It's single-stranded. It has a ribose sugar instead of deoxyribose, and it swaps out thymine for a base called uracil.
So U replaces T?
Right. But the most vital difference is mobility.
Oh, because DNA is stuck.
Yes, DNA is locked safely inside the nucleus, or the mitochondria, and the chloroplasts for plants. But RNA gets to travel. It moves out into the cytoplasm and heads to ribosomes.
Here's where it gets really interesting, because I was trying to picture how this actually works inside a microscopic cell, and I realized it's basically a high-end restaurant operation.
Oh, I like this. A restaurant operation?
Hear me out. Think of it like this. DNA is the master reference book locked in the vault of the library, which is the nucleus.
Okay.
You absolutely cannot take the master copy out. It's too valuable. If it gets damaged, the cell dies. So transcription is the process of going into the library, unzipping that weak hydrogen bond zipper just enough to find the exact page, and making a quick, single-stranded photocopy of one specific recipe.
And that photocopy is the messenger RNA.
Exactly, the mRNA.
I like this visualization. And then what happens to the photocopy?
You take that mRNA photocopy out of the library vault and walk it over to the kitchen.
Which is the ribosome in the cytoplasm.
Yes. Now you're in the translation phase. You hand the recipe to your sous chefs. And the sous chefs are the transfer RNA, or tRNA.
Right, they do the heavy lifting.
They read the recipe, and they know exactly which ingredients to fetch from the pantry.
The amino acids.
Yes, the ingredients are the amino acids. The sous chefs bring the exact right ingredients to the counter, line them up in the perfect order, and cook the dish by forming peptide bonds between them, and boom, you have a fully functioning protein.
What's fascinating here is the sheer mathematical precision required to make your restaurant analogy work.
How so?
In your kitchen, the tRNA sous chef has an anticodon, a sequence of 3 letters that must perfectly match the 3-letter codon on the mRNA recipe.
Oh, so it's a verification step.
Exactly. It's a microscopic lock and key mechanism. If they interlock perfectly, the exact correct amino acid is added to the chain. If they don't, the protein fails.
It's just mind-blowing that this is happening millions of times a second inside us.
It really is.
But, you know, I was so relieved to read a very specific instruction in the guidelines regarding this. It explicitly states, quote, Names of specific codons, anticodons, and their amino acids are not to be memorized.
Yes, the profound pedagogical choice.
It saves so much brain ache.
Well, the curriculum designers are saying, we don't need you to act as a biological dictionary. They want the students to grasp the unbroken chain of information transfer.
The actual system.
Yes, they are testing the mechanism of life. They want you to understand how W. the sous chef reads the recipe, not to force you to memorize every single recipe in the book.
That makes total sense. But it brings up a huge conceptual hurdle for me.
Which is,
if DNA is this perfect, locked-away master recipe book and we just keep transcribing it, how do we pass it on to the next generation without just creating a planet full of exact, identical clones of ourselves?
That brings us to the engine of variation.
Meiosis.
The process of meiosis.
It's a massive chunk of the curriculum, 21 marks, and it's a whole different ball game from simple cell division.
Right, simple division is mitosis, where a cell just copies itself.
To understand how we don't end up as clones, the students first have to grasp the difference between somatic cells and gametes.
Correct.
Okay.
Somatic cells are our regular body cells, skin, muscle, bone. They are diploid, meaning they have the full set of chromosomes.
Which in humans is 46.
Exactly. Gametes, on the other hand, are the sex cells, sperm and ova.
And they are haploid.
Meaning they have exactly half that number, 23.
So meiosis is this complex mechanism of taking a diploid cell with 46 chromosomes and somehow reducing it to a haploid cell with 23.
And it happens in two main phases, meiosis the fourth and meiosis the second. It starts with DNA replication in interphase. Imagine single threads of DNA cocking themselves so they look like little X's, held together in the middle by a knot called a centromere.
But the magic really happens in the very first stages, in prophase I and metaphase I, because earlier I asked why we aren't all clones. The guidelines highlight two specific mechanisms that guarantee every single gamete is entirely unique.
Let's hear them.
First is crossing over in prophase I. This blew my mind. The chromosomes literally line up next to each other, physically entangle, and swap pieces of genetic material.
They trade chunks of DNA.
Just mix and match.
And the second mechanism is the random arrangement of chromosomes along the equator of the cell during metaphase I.
Okay, visualize that for me.
Imagine all those chromosomes lining up down the middle of the cell like dancers on a stage, getting ready to be pulled apart to opposite sides.
Right.
The way they face which side of the cell they get pulled toward is completely random every single time a cell divides.
This is why you have your mother smile, but you don't have her height. It's because the gamete that created you was a completely randomized shuffled deck of your grandparents' genes. And the exam asks students to understand the mathematical necessity of this too. They call it the halving effect.
Yes. Meiosis halves the chromosome number specifically so that it can overcome the doubling effect of fertilization.
It is an elegant mathematical solution to a biological problem.
Because if a sperm and an egg both had 46 chromosomes,
their child would have 92.
and the next generation would have 184.
It would be biological chaos. The cells would be so bloated with DNA they couldn't even function.
Precisely. By having it to 23, the math works perfectly.
23 plus 23 gets you right back to a constant 46 chromosomes, generation after generation.
It's a beautifully balanced cycle.
But, I mean, I have to push back on this design a little bit.
Okay, why?
Looking at these guidelines, the sheer complexity of meiosis the first and meiosis the second, there are so many phases: prophase, metaphase, anaphase, telophase, and then you do it all over again in phase 2.
It is a lot of steps.
Why go through this incredibly complex, highly risky two-step division where so many things can go wrong just to mix up the genes? From a pure efficiency standpoint, wouldn't cloning just be safer?
Well, from an evolutionary perspective, cloning is actually the highest risk strategy imaginable.
Wait, really? How?
I called meiosis the engine of variation earlier because variation is the fuel for survival. If an entire population is genetically identical, a single new virus or a sudden shift in the climate could wipe out the entire species in one generation.
Oh, because absolutely no one would have a natural genetic resistance.
Exactly. The risk of errors during the complex dance of meiosis is the price paid for incredible adaptability. That constant shuffling of the deck guarantees that at least someone in the population might have the genetic quirk needed to survive a new threat.
That is a profound trade-off, risking the individual process to protect the entire species.
It is.
And the exam doesn't shy away from what happens when that individual process does go wrong. There's a whole section on abnormal meiosis.
Specifically, they require students to understand a phenomenon called non-disjunction.
What exactly is that?
Remember those chromosomes we visualized lining up on the equator?
Yeah, the dancers on the stage.
Right. Non-disjunction is when they fail to separate properly when being pulled apart in anaphase. Both copies get pulled to one side.
Ah. And the guidelines specify looking at non-disjunction at chromosome position 21 in humans.
Yes.
If that fails to separate, you end up with an abnormal gamete that has an extra copy of chromosome 21. So instead of the normal 23, it has 24 chromosomes.
And if that 24 chromosome gamete fuses with a normal 23 chromosome gamete during fertilization,
the resulting cell has 47 chromosomes.
yes. And the curriculum explicitly links this cellular error to Down syndrome.
And this is just a vital part of scientific education, isn't it?
Absolutely. By highlighting Down syndrome, the exam helps learners connect these highly abstract, microscopic cellular mechanics to real-world human conditions.
It demystifies it.
It does.
Yeah.
It demystifies differences in human development. It teaches them that chromosomal variations are a natural consequence of the very system that keeps our species adaptable. It grounds the science in empathy and reality.
Man, that is powerful. So we've seen how one single cell divides to create this incredibly unique, highly resilient genetic half-blueprint.
Right.
But how does that microscopic half-blueprint actually find its match in the real macro world? That's where we move from the cellular scale to the physical animal.
Shifting focus to paper 1.
Yes, looking at reproduction.
The curriculum covers reproduction in vertebrates and then transitions into a massive section on human reproduction.
And I really want to talk about the exam weighting here because the contrast is staggering.
It is very noticeable.
Reproduction in vertebrates, which covers basically the evolutionary reproductive history of every animal with a backbone on Earth, is worth 8 marks.
8 marks.
Human reproduction is worth 41 marks.
It is a deliberate, highly intentional imbalance. But before we get to the human element, what do those 8 marks for vertebrates actually cover?
It's all about understanding the different strategies animals use to maximize their chances of success in different environments.
So not just listing animal parts.
No, it's understanding the why. So students have to compare oviparity, which is egg laying, ovipary, where eggs hatch inside the mother for protection, and vivipary, which is live birth.
They also look at fertilization strategies.
Yeah, they look at why animals in the ocean can rely on external fertilization, just releasing gametes into the water, while land animals require internal fertilization so the gametes don't dry out.
They also touch on the amniotic egg and precocial versus altricial development, whether the young are born helpless or ready to run.
Right. 8 marks for the vast diversity of vertebrate life strategies.
And 41 marks for the human anatomy and its underlying mechanisms.
41 marks. And it goes deep. It doesn't just list the male and female systems, it explains the logic of their design.
For example.
Well, students learn that the testes are housed outside the body in the scrotum, not by accident, but because spermatogenesis requires a temperature slightly lower than the core body temperature to function properly.
Form follows function.
Exactly. They learn about the fallopian tubes not just as static pipes, but as active pathways lined with microscopic hairs called cilia that physically sweep the ovum toward the uterus.
It's incredibly dynamic.
But the part that really blew my mind was the comparison of gametogenesis, how males and females actually make their gametes.
This is where the cellular division we discussed in meiosis is applied directly to human biology.
Yes, and the two strategies are completely, totally opposed. I had never thought about it this way.
Break it down for us.
So what does this all mean? In males, it's called spermatogenesis. Diploid cells in the testes undergo meiosis, and one parent cell divides to produce 4 functional haploid sperm cells.
It was a volume game.
Mass production. Millions and millions are produced constantly. But in females, oogenesis is an entirely different philosophy.
It begins with a chemical signal from the brain.
Exactly, driven by a hormone called FSH follicle stimulating hormone, which acts like a biological starting gun. Inside the ovary, one cell inside a follicle enlarges and undergoes meiosis. It produces 4 cells, just like in males, but only 1 survives.
3 of those cells, called polar bodies, essentially sacrifice themselves.
They degenerate. The body literally trashes 3 out of the 4 potential cells.
To what end?
It does this just to consolidate all the cellular resources, all the nutrient-rich cytoplasm, into one single, massive, mature haploid ovum in a monthly cycle. It's a biological VIP treatment.
That's a great way to put it.
The biological investment in that single female egg is astronomically higher than the investment in a single mass-produced sperm.
This raises an important question
Mm,
about how we teach science.
yeah.
Why does the curriculum heavily weight human reproduction spending, 41 marks on these profound biological investments, the intricate anatomy, the hormonal cycles driven by testosterone and estrogen, compared to just 8 marks for the general vertebrate strategies?
Because it's about them, the teenagers taking the test.
Precisely. The curriculum intentionally grounds these massive, sweeping biological concepts in the students' own bodies.
That makes so much sense.
When they learn about oogenesis and spermatogenesis, they aren't just learning abstract textbook terms. They are learning the incredibly complex machinery operating inside them at that very moment.
It makes the science deeply personal.
Highly relevant.
Yeah.
And it radically demystifies the biological realities of adolescence and adulthood.
It gives them ownership over their own biology. So, when we started today, we were just looking at the meta rules of a test.
150 marks the cognitive level.
Right. But when you actually unpack the details, you see the incredible story this curriculum is trying to tell. It takes a 17-year-old student, unlocks the DNA library to show them the fundamental code of life.
Takes them through the risky, beautiful dance of meiosis that guarantees they are an entirely unique human being.
And then shows them the massive biological investment their body makes to ensure the survival of our species. Looking at a syllabus isn't just about passing a test. It's a window into how we literally pass down the manual for human existence to the next generation.
It is the blueprint for understanding the blueprint.
I love that.
But I want to leave you with a final thought looking forward.
Okay, lay it on me.
We discussed the 5% of the exam dedicated to level 7 learners, the highly complex synthesis questions, and we discussed specific Aim 3, emphasizing the relationship between science, history, and the lived environment.
Right.
Given how rapidly our understanding of genetics is evolving, particularly the exploding field of epigenetics,
Remind me, epigenetics.
it's the study of how our behavior, our stress, and our environment can cause chemical changes that affect the way our genes actually work.
Oh, right.
So, considering that, what will those level 7 questions look like in 20 years?
Oh, wow. That
Yeah.
changes the whole paradigm.
Will the students of the future be tested, not just on how the DNA master copy is transcribed, but on how our environment, our nutrition, and our cultural history literally rewrite the margins of our genetic code?
That's wild.
Will they be tested on how the lived experiences of their ancestors chemically altered the very blueprints they were born with?
That is an incredible thought to end on. You are your DNA, but your DNA is also a living record of everything your ancestors survived. Thank you so much for joining us on this deep dive. Wherever you are, take a moment today to ponder the incredible, tireless biological machinery running inside you right now.